Hi dear students, welcome to S&S Bioinfo. Today's topic is chromosomal theory of inheritance. Gregor Mendel conducted several hybridization experiments on garden pea or Pisum sativum to study the inheritance of genes. He conducted monohybrid crosses, dihybrid crosses and so on, and finally he made observations based on those crosses. Then he proposed the principles of inheritance. Those are the law of dominance, the law of segregation and the law of independent assortment. Gregor Mendel published his work in 1865, but it was unrecognized till 1900 because of several reasons. He published his works in a local journal. He was not a scientist. At that time, the biological world was very busy with the natural selection theory of Charles Darwin. Besides, Mendelian mathematical derivations for explaining biological concepts was unacceptable to the contemporary scientist, so that his works unrecognized till 1900. In 1900, three scientists named De Vries, Correns and Schermak redid and rediscovered Mendelian pattern of inheritance. Who are the scientists? De Vries, Carl Correns and Schermak. In 1900 rediscovered and redid the Mendelian experiments. They got the same conclusion as that of Gregor Mendel's observations. At that time, Mendelian period, the microscopy was not advanced. Gregor Mendel was unable to provide the physical proof for his factors. We have already learned about his factors. According to him, characters are controlled by a pair of discrete units called factors. Factors inherit from parents to offspring. That means they transmit from generation to generation. But he couldn't give physical proof for the factor, or actually what is that made up of. Then later, due to the advancement in microscopy, scientists could observe the movement of colored bodies during the cell division. Those colored bodies were visible by proper application of stains. Those colored bodies were known as chromosomes. Chroma means colored, soma means body, chromosomes colored bodies. They could observe the movement of chromosomes during mitosis and meiosis. By 1902, two scientists named Walter Sutton and Theodore Boveri noted that the behavior of chromosome is parallel to the behavior of genes. The two scientists were Walter Sutton and Theodore Boveri in 1902. So they noted that they observed parallel behavior of both chromosomes and genes. Thus they proposed the theory of the chromosomal theory of inheritance. According to this theory, the Mendelian factors or genes are located on the chromosomes, which show segregation and independent assortment during cell division. Now let's recall the theory of Gregor Mendel to correlate the same with the chromosomal theory of inheritance of Sutton and Boveri. This law of dominance states that characters are controlled by a pair of discrete units called factors. So here factors occur in pairs. Here first point, factors occur in pairs. Factors or genes occur in pairs. Second point, the law of segregation states that alleles segregate during the meiosis. So, alleles segregate during cell division. Third point, the law of independent assortment. Alleles or factors assort independently. Alleles or factors assort independently. When we think about the chromosomal theory of Sutton and Boveri, then according to them, the chromosomes occur in pairs. Those chromosomes carry the genes or factors. Chromosomes which have these genes or factors occur in pairs. Chromosomes which have genes or factors occur in pairs. Second point, homologous chromosome. These chromosomes are paired homologous chromosomes. Homologous chromosomes segregate during meiosis. Homologous chromosomes segregate. Third point, chromatids assort independently. As chromosomes assort independently, chromatids assort independently, the alleles also assort independently. So, chromosomes assort independently. Here, you can observe the parallel behavior of both genes and chromosomes. Genes occur in pairs, chromosomes also occur in pairs. Genes or alleles segregate each other during cell division. Homologous chromosomes also segregate at a time of cell division. Alleles or factors assort independently. Here, chromosomes assort independently during cell division. So, these are the parallel behavior of both genes and chromosomes observed by Sutton and Boveri. Now, I am going to give an explanation on the chromosomal theory of inheritance with the help of a diagram. In this diagram, you can see the meiosis stage, the germ cell formation in a cell with the 4 chromosomes. You have heard that meiosis is a cell division. It's a reduction division. Meiosis 1 is the reduction division. Meiosis 2 is same as that of mitosis. It's an equational division. Reduction division means in reduction division, chromosome number becomes half. In equational division, the same copy of chromosome will be produced in the daughter cells. Here you can see the different stages of cell cycle. So here G1 is the beginning of DNA replication. DNA replication means the DNA will be duplicated. The genetic material will be duplicated so that you can see the completed DNA replication in the G2 phase. Successful completion of the DNA replication take place in the G2 phase. So here you can see the chromosomes. So this homologous pair of chromosomes are in duplicated structure, that is bivalent structure. Bivalent or tetrad. So here you can see 2 pairs of homologous chromosomes. So what happens during meiosis 1 and anaphase? So these homologous chromosomes segregate each other. They move towards the opposite poles of the cell. Then the next phase is meiosis 2. Meiosis 2 is equal to mitosis, that's the anaphase phase. So here what happens? The segregation of these chromatids, the sister chromatids of each chromosome move towards the opposite poles. So here you can see these 2 sister chromatids move to the opposite poles. These 2 also move to opposite poles. Here also you can see the same pattern. So this results in the formation of 2 copies of cells. Here that one cell with the same genetic material, then next one with genetic material, then third, fourth. Here you can see the haploid set of cell. Haploid cell means here the chromosome number is half. What is the initial chromosome number? 4. Here it is 2. So that is the germ cell formation. So germ cells are haploid in nature because reduction division takes place during meiosis 1. And equational division takes place during meiosis 2, that leads to the formation of 4 haploid germ cells from a diploid cell. In the previous diagram, you could observe the segregation of chromosomes and chromatids during the germ cell formation. Now you can look at the independent assortment of chromosomes, the diagrammatic representation of the chromosomes, the independent assortment of chromosomes. So you know that anaphase 1 and anaphase 2. What happens during anaphase 1? Segregation of chromosomes, and anaphase 2, then segregation of chromatids. Here you can see the two possibilities how the chromosomes assort independently. So look at the possibility 1 and the possibility 2. So here you can observe the 4 chromosomes with the 4 different colors. Here this is one pair chromosome homologous, this is another set of homologous chromosome. So possibility, let us discuss about possibility 1. So 2 pairs of homologous chromosomes. Here anaphase 1, that what happens? The segregation of homologous pair of chromosomes to opposite poles. Then meiosis 2, what happens here? Then segregation of chromatids towards the pole, that results in the formation of 4 haploid germ cells. Here you can see after the segregation of the chromatids, so this is a haploid cell, second haploid cell. This is also the copy of these. These are the copies of this particular cell. So the chromatids segregate each other to the opposite poles and results in the formation of 2 daughter cells. Here you can see the combination of the long chromosome and short chromosome. For easy identification, I have drawn the long chromosome and short chromosome. See this that result in the haploid set of chromosome in the daughter cells. So the diploid set, then haploid set. Then 2 pairs, 1 pair. That is meiosis. Here possibility 2. So instead of this pair, I have used separate colors. See here red with green, here red with the bluish color. Here orange with the blue, here orange with the green. Another possibility. So that means the chromosomes assort independently. Homologous chromosomes segregate each other. The chromosomes assort independently. So here another possibility is the meiosis 2, segregation of chromatids result in the formation of 4 haploid germ cells. Here also you can see the combination of the chromosome. So here it was with the red and green, here it is with the red and blue. So here is the orange and blue, here orange with the green. So similarly, independent assortment of chromosomes take place during the gamete formation. So that Sutton and Bovary argued that segregation of chromosome take place during meiosis that results in the separation of alleles that are present in the chromosomes. So they correlated this theory with the Mendelian concepts of inheritance and finally proposed the chromosomal theory of inheritance. With this, I conclude my session. I hope you understood the topic well, the chromosomal theory of inheritance. Thank you and have nice moments.